In this video, we're going to learn about proteins. We're going to learn about their structure, how they're combined together, and what happens to them when they're exposed to heat or chemicals. So first of all, proteins are what DNA codes for. The purpose for DNA is to carry all the instructions for building all the proteins within our body and within each cell. Proteins make up 50% of the mass of all of our cells, and they have both a structural and a functional role. So some proteins have a structural role. So like keratin makes up our fingernails and our hair. And then some proteins have a functional role. So enzymes are functional proteins in that they perform reactions in our body. So they speed up reactions in our body without being used up themselves, so they're known as biological catalysts. Proteins are amino acid polymers, so that means that their subunit is an amino acid, and they just are repeated over and over and over again. And there are 20 amino acids, and 8 of them are said to be essential amino acids, meaning that our bodies can't build them from building blocks. We actually have to get them fully in our diet. So we'll just go over a few examples of proteins. First of all, enzymes, as I mentioned before, are biological catalysts. Immunoglobulins are antibodies, help fight disease. Hemoglobin, which you've probably heard about before, is a component of red blood cells. It's a carrier molecule for oxygen. Keratin, like I said before, is a component of fingernails, hair, and hooves. Fibrin is found in our blood. It's produced in a chain reaction to help clot blood, prevent blood loss. And then collagen is part of our bones and skin. Now, amino acids can be polar, nonpolar, acidic, or basic, and that all depends on one part of their structure, and that's their R group. So if we just look at a amino acid representation here below, there's 3 parts to note. So first of all, the one end has an amino group. So NH2 is an amino group. That's where the word amino acid comes from. And then the other component at the other end is a carboxyl group. So this is a carboxylic acid, so amino acid. In the middle is a second carbon right here. And on that, there's what we call an R group. That's the side chain on the amino acid. And there's 20 different side chains for the 20 different amino acids. The simplest side chain is H, which makes the amino acid glycine. But these can be quite intricate. There could be a ring. There could be a long carbon chain or short carbon chain. There could be lots of different components there. So that part is what makes it polar, nonpolar, acidic, or basic. Also, the 3D structure of a protein depends on the amino acid sequence, so the order of amino acids that are placed in the protein. So because there's 20 different ones, there's so many different combinations. And proteins can be any different length, so as short as a few amino acids to as long as several thousand. So again, depending on the order, there will be interactions with the different parts of the polypeptide chain, and it will cause it to fold into these intricate 3D shapes. To build proteins, amino acids are joined together through condensation reactions, and the bonds that hold them together are called peptide bonds. And there's 4 levels of protein structure that we'll talk about, but first we'll take a minute to look at the peptide bond. So what happens is 2 amino acids line up. So they line up at their carboxyl and amino regions. And the OH comes off of the carboxyl group, and one of the hydrogens comes off the amino group, and then you end up with 2 amino acids joined together, or a dipeptide. And I didn't mention this before, but one way to help you remember the structure of an amino acid is to look at the center part. And if you look at it, the center part makes NCC, so the initials for our school. So that helps me remember the order in which you write an amino acid. So the first level of protein structure is the primary structure. So that's just the amino acid chain. So the DNA would include instructions on how to build the protein, and the amino acid chain would be built from that. So we have this one, we have met, so methionine, leucine, glutamic acid. It just keeps on going. DNA, like I said, has the directions for building that protein. So this is before any interactions have taken place amongst the members of the polypeptide chain. So that's primary structure. So we're going to spend a little bit longer time on secondary structure because there's two different types of hydrogen bonding that can occur between the carboxyl and amino regions. So in secondary structure, there's hydrogen bonding between the carboxyl and amino groups. If you remember, the amino group has a hydrogen, and then the carboxyl group has a carbonyl. So there'll be hydrogen bonding between the hydrogen of the amino group and the carbonyl on the carboxyl group from adjoining or nearby amino acids in the polypeptide chain. And they can either form alpha helices or beta pleated sheets. We're going to spend some time looking at each of those. So the first type of secondary structure is alpha helices or an alpha helix, and that's when hydrogen bonds form between the amino region of one amino acid and then the carboxyl region 4 amino acids away. So if we go, this is amino acid 1, 2, 3, 4, and then there's a hydrogen bond, and that you can see it happening here to this one over there, which is 4 away, this one to this one, which is 4 away, and so on. Because there's that continuous and regular interaction, so between a carboxyl and amino 4 away, you end up with this coiling pattern, so it forms a helix. So an example of a protein that has a lot of alpha helices would be keratin, which is found in hair. The second type of secondary structure is beta pleated sheets. So what happens is two parts of the polypeptide chain fold in a line parallel to each other, and hydrogen bondings occur just adjacent. So the carboxyl group of one section right here to the amino group on the next or parallel part of the chain. So you can see that all the way along there's a regular hydrogen bonding pattern, and this would be seen in spider silk. So spider silk has a lot of beta pleated sheets. So tertiary structure is when we say the polypeptide has supercoiled, and that's because all the side chains start interacting with each other and with the environment around them. So you get these crazy 3D structures. So some examples of side chain interactions might be hydrogen bonding between, a hydrogen and an oxygen in adjacent parts of the polypeptide chain, or ionic bonding. So you end up with an ion here and an ion there. One's positive, one's negative, so they attract. Another one, disulfide bridges. So there's a sulfur that forms a covalent bond with another sulfur in another amino acid. And then finally, nonpolar regions will interact with each other. So these are nonpolar because they're methyl groups, and they just want to hang out together and want to avoid water. The final level of protein structure is quaternary structure, and that's when two or more proteins or polypeptides fold together amongst themselves to become that functioning protein. So, for example, hemoglobin has 4 subunits, so there's 4 different polypeptides that fold on each other, and you end up with these really unique 3D shapes, and that's quaternary structure. So proteins are so diverse. They can be as small as 1 amino acid to be a functioning protein, and they can be as long or as large as thousands of amino acids combined. The longest amino acid structure, so the longest protein, is known as titin, which is over 39,000 amino acids long, I believe, and it's found in our muscles, and it gives us the springiness to our muscles. The 3D shape of a protein, like I said before, depends on that amino acid sequence. And if we get an error in the DNA, we will get an error in the amino acid sequence, and then the protein may not fold properly, and the protein can't fold properly, it won't be able to function properly. So we'll spend a little bit more time about that when we talk about molecular genetics in a later unit this year. Amino acids can be combined in any sequence, so that allows them to have any number of different protein possibilities because they'll fold in different ways if you put them in different sequences. Finally, we'll take a look at denaturation, and this is when a protein gets damaged. So we call this the unfolding or unraveling of a protein. So what happens here is all those interactions between parts of the amino acid chain get broken. So the hydrogen bonds might break between the carboxyl amino groups, so the secondary structure might get damaged. Ionic bonds or disulfide bridges can be broken, so the tertiary structure could be broken. So it's just unraveled. But we don't actually lose the polypeptide chain order, so the primary structure still remains intact. But in denaturation, it could occur because of heat. That's why we need to make sure that we don't have high fevers in our bodies. And it's also why when we heat proteins like meat and eggs, they change in their conformation because we have denatured them. And we've actually gone even further and coagulated them. So pH outside the optimal range can also do this. So really low pH or really high pH can damage a protein. And at that point, the protein is no longer functional because the 3D shape determines its function. And so if it doesn't have that shape, then it can't function properly. And finally, if the peptide bonds between the amino acids actually get broken, that would be the primary structure being damaged. The protein is said to be permanently damaged, and we say that is coagulation. And you can't reverse coagulation, but sometimes you can reverse denaturation, because if you remove the protein from the heat, the refolding can possibly happen again.